Hello, dear students. Welcome in the lecture series on genes and chromosome. I am Professor Hemlata Suryawanshi, Assistant Professor in the Department of Microbiology, Pratap College, Amalner. Today we are going to study one of the most important concept in molecular biology, that is genetic code and its properties. So, before understanding genetic code, recall central dogma of molecular biology presented by Francis Crick. So you can see in this image, there is a DNA. Let me take a pointer here. Yes. So there is a DNA. Genetic information stores in the DNA. DNA transcribed into RNA. And as we know that RNA carries that information, messenger RNA is there. Then by translation, RNA codes certain proteins. So protein expresses that information. See, information, genetic information stores in DNA. That information carried out by RNA and then protein expresses that information. So one of the question arises in the mind. How does sequence of DNA bases decides which amino acids will form a protein? The answer lies in genetic code. So now, genetic code. It is the sequence of nucleotides in DNA or RNA that determines amino acid sequence of proteins. Or we can also say that genetic code is the set of rules by which the sequence of nucleotides in mRNA determines the sequence of amino acids in the protein. This genetic code, this term given by George Gamow. Then codons, one another concept. Codons are the group of three adjacent bases, or we can say group of three adjacent nucleotides that specify amino acids of the protein. mRNA is read in the group of three nucleotides. Each group of three nucleotides forms a codon and one codon codes for one amino acid. You can clearly understand from this table. This is a table of genetic code. See, singlet or doublet codes which are not adequate to code for 20 amino acids. Okay. Therefore, triplet code, minimum triplet code is required. 64 codons by taking this type of arrangement, as we understand that 64 codons are formed which codes for 20 amino acids. They also possess start and stop codons. In the triplet codon, we can say triplet codon. So triplet codon, 3 RNA bases codes for 1 amino acid. See the arrangement. The nitrogen bases U, C, A, G, uracil, cytosine, adenine, guanine. So this these are the first letter which we take in a codon. Then we take second letter from here and the third letter from this sequence. So you can see this U, then second letter here again U, and then third letter from this side. So U, that is the triplet codon. Then again we take this U, but this letter. So U and C. So in this way, 64 codons are formed, and these 64 codons codes for 20 amino acids. And among them, start codons are there, which we call them as initiation codon. So example is AUG. This codes for methionine in eukaryotes and formylmethionine in prokaryotes. This initiation codon initiate protein synthesis. So, in other way, we can say that it tells the ribosome that start reading from here. So, start codon is AUG. Now, the stop codon. When protein synthesis is terminated, then stop codons signals give signals, which we call them as a termination codon. There are three stop codons: UAA, UAG, and UGA. These stop codons, as they stop, they terminate the process. They do not code for any amino acids. They are giving signal for termination to stop the process. These were identified in experiments by Marshall Nirenberg and Hargobind Khurana. Now, the most important concept is a Wobble hypothesis. See, there are more than one codon for one amino acid. This is known as a degeneracy of a genetic code. One amino acid codes for more than one codon, or more than one codon for one amino acid. So, to explain the possible cause of degeneracy of codons, in 1966, Francis Crick proposed this concept known as a Wobble hypothesis. So, let me explain this in detail. According to this Wobble hypothesis, only the first two bases of codons have a precise pairing with the bases of the anticodons of tRNA. See in this image, these codons are present. This is the triplet codon present on mRNA and complementary anticodons present on tRNA. So, this tRNA which carries leucine amino acid having the anticodon GAG, this is the identical leucine tRNA which also contains GAG. But see, they are complementary with the codons on mRNA CUC, and similarly this third nitrogen base, it can pair with you also, isn't it? So while pairing between the third basis of codon and anticodon, there may be wobble. Wobble means what? Not steady, unsteady. This phenomenon permits the single tRNA to recognize more than one codon. And therefore, although there are 61 codons, 61, because 3 codons are the stop codons, that's why 61 codons are for amino acids. The number of tRNA is far less. It is around 40. Why? Due to wobbling. Then we move towards the properties of genetic code. The first property is a triplet code. Codons are always in a triplet form. Each codon consists of 3 nucleotides, hence triplet. You can see here, this is the RNA and the sequence of bases CUC3, they form one codon. Then again next CGG form the another second codon. Then AGC forms third codon. So in this way there are three bases in a codon, hence known as a triplet codon. Now non-overlapping nature, it is the second property. Codons are read one after another without overlapping. For example, see here, AUG, UUU, GCU. You can see in this image, this is the mRNA. If the overlapping is there, then this type of overlapping codes we can observe, but it not happens. So there are three codons, then next three, then next three. So in this way, overlapping avoided. Then comma-less nature. There are no punctuation marks between codons. The sequence is read continuously from start to stop. No any break, no comma, no full stop. For example, A U G U U U G C U A A G, like this. Cell automatically reads it in the correct frame. In the image, you can see that this is the first codon AUG. There is no break, no comma. Again, the second codon CAU. So continuously we can read the frame. Then fourth property is a degeneracy of genetic code. See, most of the amino acids are coded by more than one codon. Most of the amino acids are coded by more than one codon. Let's say example, leucine coded by 6 codon. You can see here in this table UUA, UUG codes for leucine, and again these CUUU, CUC, CUA, CUG, they are also codes for leucine. So there are total 6 codons. Here this amino acid coded by these or we can say the single codons are there, they code the same amino acid, isn't it? Then serine, serine, 6 codons. You can see here AGU, AGC codes for serine. Again here also these 4 codons, they code serine. Then glycine, see this amino acid which is coded by 4 codons. Uh, See here, these 4 codons are there. So this is known as a degeneracy. Many of the amino acids or the amino acids coded by more than one codon, this property is known as a degeneracy. Here, important note is that even though there are 64 codons, but only 20 amino acids exist. So degeneracy mainly occurs at the third base position, as we studied in a Vobel hypothesis. Now the next property is unambiguous nature. Each codon codes for only one amino acid, isn't it? See, UUU codes for phenylalanine. No other UUU combination is there, isn't it? So each codon codes for one amino acid. UUU always codes phenylalanine. Never code for another amino acid. You cannot observe this triplet codon again in this table. One codon, one amino acid, isn't it? But one amino acid, for one amino acid, there are many codons. For phenylalanine, UUU and UUC, we observe here. So, this is degenerate. We can say this is a degeneracy is there. And this type of phenomenon is known as a unambiguous nature. Then the last property is a universal nature. The genetic code is almost universal. For example, uh, in humans, in bacteria, plants, animals, all use nearly the same codon table, and this suggests that all organisms evolved from a common ancestors. Exceptions are also there, mitochondria, some protozoa, but these are the minor variations. So these are properties of a genetic code. So thank you. Here we concluded our lecture series on genes and chromosome.